All right, so here's an animation to demonstrate how that dynamine action can lead to these microtubule doublets to bend. I'm going to start it halfway through here to get to the punchline here, but here we have a couple of um, microtubule doublets, and here we have dynin in between, and one end of it can walk against the other. And I'll give you a more precise example of it in a few minutes here. But notice that as they start to walk along, it causes the tubes to start bending against each other. So here we go. We'll get started here. There we go. And this activity is governed by ATP hydrolysis. So we burn ATP, these dynin molecules walk along the surface, and it causes these microtubules to bend. So first of all, let's think about how dynin actually works. And here's a really helpful video looking at the molecular structure of dynin. And what's important here is that we have two arms that have the ability to walk against each other. So here we have the microtubule, here we have dynin, and it has two arms. Each head is capable of binding the tubulin surface, and in addition, ATP will cause this to shift forward. This allows two things to walk against each other. Now, something that's important here is we have protein crosslinks that will limit the microtubules from sliding too far away from each other. So in combination, that is going to cause the overall flagellum or cilium to curve or bend, leading to the activity we've seen in both sperm and in flagella. Now, that is not the only molecular motor we have in the cells. We also have actin that can work as in a motor type of scenario. So microfilaments, which are made up of actin, produces kind of a twisted double chain of actin subunits. These are not very good at resisting bending or squeezing, but they do bear tension, so they handle pooling. Now, we have a layer of microfilaments or actin filaments that sits just underneath the membrane. So here is a fluorescent image of actin that is stained. So it's glowing green, and you can see that it's sitting directly underneath the surface. Here's an electron micrograph of that structure. You can see it forms kind of a web-like structure. Here's a cartoon of that, showing how we have these actin filaments. We have various cross-linking elements which lock these into a net. And then we also have this thing called myosin, which has the ability to walk along actin in much the same way that dynin walks along microtubules. Now, we see actin show up in a lot of different ways. So here is some different examples. So as I said before, directly under the cell surface, there's this layer, a net just underneath the membrane called the cell cortex. Here's a different shaped cell, what it might look like there. You can see here that this particular cell type has these little protrusions. These are called microvilli, and they are made up of these these actin filaments that are heavily cross-linked together in a parallel fashion. That allows them to kind of push and extend out. Here we have kind of a belt that wraps around the side of the cell, creating a kind of reinforced structure. Actin is in much a similar way that we see these microvilli here, can lead to parts of amoeboid cells kind of reaching out or even pushing out the entire leading edge of a cell. In addition, we can have these things called stress fibers, which run along the surface and can actually lead to the cell being pulling in the backside because of these motor molecules running along it, these myosin molecules that can go. This can also lead to how cells divide into two. There's actually kind of a belt, similar that we see up here, that wraps around the surface and then squeezes in on itself, which actually leads to the separation of two cells. This next image here, we have an electron micrograph. We can see the plasma membrane on the outside, and then inside we can see these microfilaments formed in the bundles. Down directly underneath, you can see this net-like structure of the cortex, which again is actin. And in this case, it also ties in with these intermediate filaments, which we'll talk about a little later. You also see actin in muscle cells. So actin, inside of muscle cells, we have these long parallel fibers of actin, and in between those actin fibers, we have these these myosin filaments that have heads on both sides. Now these arms work in much the same way that dynin does and can walk along the surface of the actin. This leads to muscle contraction. Here we have an example of an amoeboid type cell. So you guys have probably seen images of amoebas crawling around. They move by sending out protrusions, or these pseudopodium, that move the cell forward. Then they can adhere to a surface, and then using contractile type of myosin and actin fibers can pull the rest of the cell in behind it. So this is extended mostly through actin polymerization. So we're kind of building fibers that get longer and longer and spread this edge. And then we have more organized fibers that will pull the tail in behind. We also see this actin activity in plant cells, where we get this effect called cytoplasmic streaming. So here is a bunch of boxy plant cells, and these green things are chloroplasts, and these cells actually move their cytoplasm within their cells to keep nutrients constantly moving. And these cells are also actively connected to each other. Here's an example of what that looks like in real life. So you can see how these chloroplasts are moving around the surface, and this is all through actin movement. Now, finally, the last part is we have intermediate filaments. Intermediate filaments are, as they are described, intermediate in size. They are very good at supporting shape and fixing, but they are static in nature. So we don't see them growing or shrinking. So, in concept 6.6, the cytoskeleton is a network of fibers that organize structures and activities in cell. Gave an example of how all these fit together, that we are 3 different types. We have microtubules, which resist bending and compression. We have actin filaments that are really good at holding tension. Both of these have motor molecules that can move along them. They can also grow and shrink. Intermediate filaments are much more static and provide the overall toughness that we see in a lot of cells, such as skin cells or what we see in hair. These microtubules are what leads to the formation of things such as flagella and cilia. And that in combination with various actin fibers can lead to a wide range of different cellular movements.